Okay, so this is lecture 20 from my genetics class, and this is sort of the second lecture on genomics. So in the first lecture, the last one we had, we focused primarily on how the genome was sequenced. And in this lecture, I'd like to talk a bit about what good is a genome sequence. So the product of sequencing the human genome is about 3 billion base pairs of sequence. But as you can see from this slide, that just having all of those strings of A's, T's, G's, and C's really doesn't tell you all that much. That somehow you need to be able to sift through this 3 billion base pairs and find the important information. And part of the important information is, of course, finding where the genes are. And once you find the genes, you can start to get into sort of higher order questions about comparing what genes are in the genome. Look at the overall structure of our genome. So where are the genes? Where are other things in the genome? We can compare our genome to other organisms, which tends to be very useful in the course of evolutionary studies. And finally, we can look at the relationship between gene expression and things like development on a genomic scale. So we have technologies now that we can identify more or less all of the genes in the genome, it is very useful to be able to look and see where all those genes are expressed and when they're being expressed in different tissues and different stages of development. So one of the first challenges of a genome sequence is identifying where the genes are. And one of the first questions you really have to do before you start identifying genes is define what a gene really is. And as this is one of those things that seems very simple, but the further you go into molecular biology and genetics is what we know of it today, it's more difficult than you would think. So broadly speaking, a gene is a sequence of DNA that encodes either a protein or a non-coding RNA. And in either case, these are sequences which are going to be transcribed from DNA to make an RNA molecule. In the case of protein encoding genes, this messenger RNA will then be translated into a protein, whereas with non-coding RNAs, it is the RNA itself which is the product that we're interested in. And some of the examples of RNAs that don't produce proteins, these so-called non-coding RNAs, some of the ones you're probably familiar with are things like transfer RNAs, which are important in the process of translation, as well as ribosomal RNAs, which make up the ribosomes. But there's also lots of other classes of small RNAs, such as small nucleolar RNAs, or SNO RNAs, small nuclear RNAs, or snRNAs, which are involved in various aspects of RNA splicing and processing, as well as things like microarrays and siRNAs, and these are involved in the regulation of other transcripts. But probably the most common thing that these things the biggest thing they have in common are the fact that they are translated into RNAs. And we'll talk later about how this is sort of the gold standard of whether something is a gene or not. Is it transcribed into an RNA molecule? So I'd like to talk about a couple of different approaches to identifying genes in the genome. We'll talk about 3 complementary approaches, and one of which deals with kind of what we just talked about. If part of our definition of what a gene is that it's made into an RNA molecule, then one way to find genes is to look at all the RNA in a cell and see where it came from, because they must have come from genes if that's how we're defining a gene. So one approach is to use these EST databases. And the EST stands for Expressed Sequence Tag. And the idea here is that you make a cDNA library. So we talked a little bit about that before. So we basically make a DNA copy of all the messenger RNAs in a cell. And then we run one sequencing run per clone. So we're not sequencing necessarily the entire transcript, because oftentimes these transcripts are going to be many thousands of base pairs in length. And typically DNA sequencing can't sequence that far in one shot. But it does give you a tag. It gives you some little bit of a label of each one of these cDNAs, and each one of these came from a messenger RNA. So one of the limitations of this sort of an approach is that you typically will not get non-coding RNAs this way. This is very good for getting messenger RNAs, but not non-coding RNAs. So why would that be? Well, it comes down to a quirk in how these libraries are made. Okay, so one of the properties of the reverse transcriptases, the enzymes that are used to convert RNA molecules into DNA copies, is the fact that, like DNA polymerases, they're not able to start reverse transcribing a transcript from scratch. So you can't just give it a free messenger RNA and expect it to start making a copy. It has to have a primer to start from. So again, like DNA polymerases, they can extend from a free 3' end, but they can't start by themselves. So this kind of causes a bit of a problem, because we need to somehow give it an end to start with, and that end, that primer, is going to have to be base paired to the messenger RNA sequence. And in the case of messenger RNAs, there's a nice feature, which is this poly-A tail, this polyadenylation that takes place on messenger RNAs. And the reason it's nice is because we know that all of the messenger RNA molecules will all have this common sequence. So we can take a whole pool of messenger RNAs, and for all of them we can use a primer that is basically just a run of T's, which will be able to hybridize to the 3' end of the transcript. And when we put the reverse transcriptase in, it'll be able to extend that and make a copy of the gene. So it's going to copy this RNA into DNA. Now, one of the reasons that this works very nicely for messenger RNAs is because of this poly-A track. But non-coding RNAs lack these poly-A tracks. So if you try to make a library in this fashion, you'll make very nice copies of the messenger RNA molecules, but not of the non-coding RNAs. So that is kind of one of the limitations of this sort of an approach. So once you have these EST sequences, you can use computers to align these to the genomic DNA. So basically you can use those EST sequences and use computers to map those ESTs onto the genomic DNA sequence. So each EST sequence, therefore, should align to one region of the genomic DNA that encodes the messenger RNA that produced it. There are some limitations, though. So one of the problems with this sort of an approach is you won't get non-coding RNAs, as we just talked about. The other thing is that the cDNA libraries only contain genes that are expressed in the type of tissue used to create the library. So, granted, all of the genes are encoded by the genome, but in any given cell type, not every gene is being transcribed into an RNA. So, there will be genes which are missing in almost any tissue type. So, if you, for instance, are looking for a gene which is only expressed, say, in your liver cell, but you made a cDNA library from, say, brain tissue, then those liver-specific enzymes would not be expressed in that brain tissue, so they simply wouldn't be there. On a kind of similar note, very rare messages are hard to find. And just to kind of give you an illustration of this, if you think about, like, if you were looking at messenger RNAs in muscle tissue, so in muscle tissue there's going to be lots of actin and myosin. So these sort of two proteins are very large components of muscle tissue. So if you were to start sequencing ESTs from this sort of a population, which is basically essentially just kind of like randomly picking out, imagine reaching into a grab bag and pulling things out, you're going to hit actin and myosin quite a lot of times. However, if there's a transcription factor which is present at very low levels, chances of pulling it out are going to be much slimmer. So there is just sort of a numbers game, that you're bound to get the more abundant things much more often than the rare things. And keep in mind that, I've drawn an example here that only has 3 proteins, but your typical cell might have 10 to 15,000 different proteins. So if you're going to just be pulling them out at random, it can take a long time until you've actually managed to pull out each of those, say, 10 to 12 to 14,000 transcripts. So, a complementary approach to the ESTs are gene prediction programs. So these are programs that scan through the genome and look for genes. So if you're going to design a program that would look for genes, what sorts of things would they look for? Well, there's a number of things. One is open reading frames. So open reading frames are just a sequence of DNA that has the potential to encode a protein. And the reason we say this is that basically you'll be able to scan along a sequence of DNA for some reasonably long period without finding a stop codon. So if you just had a completely random sequence of DNA, how often would you expect to encounter a stop codon just as you walk along it? And basically there's 3 different stop codons. So out of the 64 possible codons, 3 of them are stop codons. So about 1 in every 21 codons, if the sequence were just random, should be a stop codon. So if you go along a DNA sequence and you find an open reading frame, i.e. what I mean by that is a series of codons that continuously encode amino acids without hitting any stop codons, and if they go for hundreds of amino acids, or maybe even thousands of amino acids without a stop, then quite likely this is going to encode a gene. So in addition to just looking for open reading frames, you can sort of look at the quality of the open reading frame. So we talked before, when we talked about mutations, the fact that the genetic code is redundant, meaning that there are multiple codons which encode a single amino acid. So in this case, CAG or CAA both encode glutamine, or a CAT or a CAC both encode histidine, and all 4 of these encode leucine. But different organisms have different preferences for which codons that they'll use. So for instance, I believe this data is from human, both of these codons encode glutamine, but instead of one being used 50% of the time and the other one being used the other 50% of the time, this one is actually used about 3/4 of the time, where this one's only used about 1/4 of the time. And same thing with leucine. There's 4 different choices, so if things were completely random, you might expect each of them to be used about 25% of the time. But CTG is used about 40% and CTA is only about 7%. So there's a serious bias in which codons get used. And this can be something that you can use to evaluate whether something's really likely to be a real open reading frame. So if an open reading frame is using mostly the codons that are preferred by that organism, say the CAG, for glutamine and the CTG for leucine, then that's a good sign that it's a real open reading frame. But if you do see that these unpopular amino acids, or unpopular codons like CTA or CAA come up repeatedly, then that might be an indication that this might be a more not a real open reading frame, but just sort of a random coincidence that you've gone a long ways without hitting a stop codon. And these sorts of programs, then, looking at open reading frames, can identify at least part of, so not necessarily every last amino acid of the sequence, but at least part of about 80% of genes. And just kind of as a sort of an interesting example, when I talk about this codon bias, how different organisms have a different biases in which codons they like to use, this can be a big problem whenever you start trying to move genes between organisms. So it's a very common sort of thing to try to express human proteins in E. coli. Things like insulin can be produced in E. coli. But one of the challenges is that humans and E. coli have very different preferences for some codons. So for instance, if you look at AGG arginine codons, humans use that codon about 11% of the time, where E. coli only uses it about 1% of the time. And similarly, CCC codon for proline, humans use it quite a bit, 20%, but E. coli only uses it about 5%. Now, why this is a problem is if you try to express a human protein that's using lots of AGGs and CCCs, there simply won't be many of those transfer RNAs around because E. coli doesn't use them much, so it doesn't produce many of those transfer RNAs. So it would be very difficult to express human proteins that contain these particular codons. So this particular product from Strategene, what they've tried to do is to change the codons that E. coli uses so that it uses more of the human codons to make it easier to express human proteins in E. coli. So we talked about EST sequences, we talked about computer databases, and one of the other big things you can do is to compare the sequences between organisms. And this is really kind of beyond the human genome sequence. So this, in 2012, we've sequenced lots of other mammals and mammals as well as different insects and bacteria and everything else. And the basic idea is that if we take two different organisms and we compare their genomes, then we can look for sequences that are the same. So I've kind of tried to draw some conservation of sequence between human and mouse, where there are sort of blocks where there are related sequences and blocks where the sequences don't show any similarity. And the idea is that since humans and mice have been evolutionarily diverged for millions of years, that if there are sequences that are still relatively the same, still similar to each other, then there must be a reason for conserving them, and that reason would presumably be because these are genes. So once we have an idea that something is a gene, say from the computer prediction programs or from conservation between two different organisms, one of the ways to verify that these things really are genes is to look and see if they produce RNA molecules. And one of the simplest ways to do this is reverse transcription coupled PCR, or RT-PCR. And this is very similar to the first step in that library construction, that we take those messenger RNA molecules, convert them into cDNAs, or DNA copies, and then these can be used as a template for PCR. And we've talked about PCR before, and you've probably had it in previous courses. If it doesn't ring a bell, you can go look it up. There's plenty of good videos and things like that on YouTube if you want to see how PCR works or need a refresher. But this will allow you to detect to see if you can actually detect a messenger RNA or a non-coding RNA that is coming from this thing that you think is likely to be a gene. And if you can detect an RNA, then it's a pretty good sign that what you've got is a real gene. Okay, so what is in your genome in the terms of genes? So I've mentioned this before in class, so somewhere around 1990, people were guessing that there might be 100,000 genes. In 2001, by the time we came out with a draft sequence of the human genome, it was estimated there were probably about 40,000 genes. And this number has come down even further. So by 2004, the complete sequence of the human genome was out, and it looks like maybe 20 to 25,000 genes. This was a little bit sobering, because, I mean, as humans, it's a little difficult not to be a little egomaniacal and think that we are the most advanced organisms on the planet, so we ought to have the most genes. And to some degree, this seems to be the case, so we have more genes than, say, fruit flies or C. elegans. Interestingly, however, we probably have less genes than Arabidopsis, which is a small plant related to mustard. We have less genes than, say, a pufferfish. And we certainly have less genes than something like rice. So, what does all this mean? So, maybe the number of genes isn't really a very good indicator of complexity, or maybe there's something more important than just numbers when it comes to genes. And we'll come back to that thought in a second. So what kinds of genes are in our genome? So I said there's maybe around 25,000 genes in our genome. We'll go on the higher estimate. So one thing that is very clear is that there are not 25,000 completely unique, unrelated proteins in the genome. So what we see instead is that some types of genes get used over and over and over again. So here's a table of like the top 20 families of proteins and genes in your genome. And together these 10 types of proteins make up about 20% of your genome. So things like immunoglobulin domain proteins, part of your immune system, the C2H2 zinc finger proteins, which are involved in DNA binding, kinases. You can see that there's hundreds of these, so 765 immunoglobulin domain proteins, 706 C2H2 zinc finger proteins, 575 eukaryotic protein kinases. So it was about 2,000 genes that are represented by these 10 classes. So what we see is that the same sorts of proteins get made over and over and over again. Now these sequences aren't exactly the same, they're slightly different. So for instance, these zinc finger proteins, which are transcription factors, are probably binding to hundreds of different sites, but they're all roughly the same sort of model. So these are lots of variations on a common theme is basically what we see in the genome. And judging from the last figure, it shouldn't come as too much surprise that there's a lot of modular construction in our genome. So we see the same domains occurring over and over again in different combinations. So again, a domain is a subsection of a protein that's going to fold up into its own little tertiary structure and have some given function. So it could be a kinase domain, which puts phosphates on other proteins. It could be a DNA binding domain. Could be a different sort of activity, sort of a glycosidase domain. And what we see is that there's a lot of modular construction. So these different domains can be put together in different ways to make proteins of different functions. So kind of think of these as sort of Legos, sticking them together. So instead of inventing completely new DNA binding proteins, what you see are variations on the same themes. So these are all different transcription factors, and you can see that in some cases these C2H2 domains get stuck with pause domains, in other cases they're stuck with crab domains, and in other cases they're stuck with scan domains. And I don't expect you to know any of the names of these domains, but try to pick up on the theme. So here's a homeodomain, that's another DNA binding domain. Again, it can be stuck with a paired domain, it can be stuck with a POU domain, it can be stuck with a limb domain, it can be by itself. So there's lots of different ways that these things kind of get taken apart and put back together in different orders. And these domain combinations, sometimes they're shared between species, and other times they can be unique. So there are particular combinations which occur only in some species. So for instance, in Drosophila and flies, we find proteins that have this pause domain with this little A domain, but we don't find that combination of domains in humans. And similarly, humans have domain structures that aren't found in flies. So for instance, this pause domain with a B-zip domain. And again, these are still all transcription factors, but this is a domain structure or a domain architecture that doesn't occur in flies. So some can be common, like this pause with C2H2, to both flies and humans have those types of proteins, but then other ones are more distinct. Now, back to this concept about humans being the most sophisticated, most advanced organisms on the planet. If you want to make that argument, one of the ways that you can make it, since it does not seem to work out very well arguing that we have more genes than everything else does, the one thing we do seem to have more of is we seem to have more unique domain architectures. So we have this modular construction in the genome, and if you look at the layout, if you look at all the different ways that those domains are sort of plugged together to make different types of proteins with different combinations of domains, this is the one area where humans do seem to distinguish themselves from other species. So the number of different unique domain combinations is higher in humans than most other species. So this may have something to do with why humans are a bit more complex than other organisms. Okay, so we talked a lot about what sorts of protein sequences are in the genome, or what sorts of gene sequences are in the genome, things that encode proteins, some non-coding RNAs. But what else is in the genome if you look beyond genes? So there's lots of repetitive sequences in the genome. So repeat sequences constitute about 50% of the human genome. So transposons account for themselves about 45% of the genome. We also have various pseudogenes. So pseudo sort of meaning false genes. And many of these have arisen by retrotransposition. So we talked about this a little bit with transposons, but occasionally DNA can get transcribed into a messenger RNA. But then what's not typical is that can get reverse transcribed back into a DNA copy, and then those occasionally will get integrated back into the genome. So one of the ways to recognize a pseudogene is the fact that it's not going to contain introns or promoters, because the pseudogenes came from reverse transcription. So the messenger RNA lacks the introns and never did contain the promoter sequences. So when this gets transferred back into a DNA and goes back into the genome, it won't contain these sequences anymore. Another major class of repeats are these simple sequence length repeats, and these are pretty much what they sound like. They're very simple sequences that are repeated. So things like dinucleotide repeats or even just single nucleotide repeats go over and over and over and over again. And we talked about these a little bit in terms of things like Huntington's disease is an example of simple sequence repeats, those CAG repeats. And these account for about 3% of the genome. Some of the other things you'll find in terms of repetitive DNA are these segmental duplications. So these are duplications of larger blocks of DNA sequence. So somewhere between maybe 10 and 300,000 base pairs, 300 kilobases, gets copied and inserted someplace else in the genome. Now these are relatively large pieces of DNA that are capable of encoding genes themselves. So one or more genes could easily fit in these 10 to 300 kilobase sections. And this makes it a very important force in the evolution of new genes. So when you think about evolution, the general idea with evolution is that you start out with a given gene sequence and we have mutations, and somehow those mutations can alter the function of the protein, either by changing the protein sequence or by changing the expression pattern of the gene, and this allows it to develop a new function. So the problem with this sort of evolution is that if the gene you're starting with is an essential gene that has an essential function that you can't live without, that if you have mutations which would give it a new function, then this can often be deleterious or even lethal, because you've lost the function of that essential gene to begin with. So you can't go mutating this copy of the gene to make a new gene without having bad effects from losing the original function of this gene. So this is why these segmental duplications are really kind of handy in the course of evolution. Because if you end up with a segmental duplication, then this gives you the opportunity to have one of those copies then maintain the original function of the gene, that original essential function, while the other copy can be mutated. And occasionally these mutations will give rise to a new function for the protein, which will allow it to evolve with a novel function. So this duplication first allows you to sort of keep a copy for the old function and mess with a copy with the idea that it might eventually give rise to a new function. So where are the genes in the genome? So we have 3 billion base pairs of DNA spread across 23 chromosomes. Are they uniformly spread across the chromosomes? No, not really. So there do tend to be very high density regions. So the region on chromosome 6, where the histoincompatibility complex is, has about 60 genes in 700 kilobases. This is about 1 gene per 12 kilobases, and this is pretty high for the human genome. This is a fairly, would be considered a high density region. But then there are also gene deserts, which contain really no genes as far as we can tell, for maybe millions of base pairs. So there is a lot of variation. One of the other things you can start to look at when you have multiple genome sequences, say mouse and human, you can start looking at genome evolution and how genomes change. And one of the concepts people talk about with genome evolution is synteny. And synteny basically means conserved gene order, so that as you go along a chromosome from, say, human and mouse, if you see the same genes occurring in the same order, then this would be synteny. And what you can see is it's sort of interesting. So this is just a partial list of the human chromosomes, so 13 through 22 in the X and the Y. And then this color coding below shows the mouse chromosomes that have the same sequences as these human chromosomes. And what you can see is that some of them are conserved very well. So for instance, chromosome 20 is almost entirely related to chromosome 2 from mouse. And the X chromosome from from humans, and the X chromosome from mice are very much the same. Then other ones you can see where there are chromosomal rearrangements. So for instance, say this chromosome here, chromosome 15 from humans, sequences from, say, chromosome 3, 9, and 2, in these three different colors, are represented on this chromosome. And it turns out that if you have about 170 rearrangements, you can take the mouse chromosomes and make a pretty good facsimile of the human chromosomes. So at this point, we have the human genome sequence. We have the sequence of all the bases, all 3 billion bases, and we have a good idea of what the genes are, so about 25,000 genes. So, are we finished? At this point, are we done with genomics? Well, not by a long shot. So, as all of you know, all of the cells in your body have the same DNA sequence, for the most part anyway. So, how is it then that different cells take on different fates and perform different roles if they all contain the same DNA sequence? So this is just a bit of an illustration showing you different types of blood cells. And these cells have very different roles, they have different shapes and morphologies, different biochemistries, and different functions in your body. So how do you get all of these different types of cells from a single genome sequence? And the key deals with the expression. So you have 25,000 genes in your genome, but not every cell is expressing the same set of those genes. So a big part of differentiation and specialization inside your body deals with which genes are being expressed in which cell types. So just to give you an idea of what is going on inside a single cell, a typical cell may be expressing somewhere between 10 and 15,000 different genes out of those 25,000. And even though they're all expressing them, so two cells which are expressing the same gene may express them to a very different level. So altogether, maybe there's 300,000 different messenger RNAs in the cell at a time. So this is a really complex mixture. And if we're going to understand something about development and how the genome is expressed and how that results in phenotypes, we need to be able to tell something about gene expression. And kind of one of the workhorses for looking at expression levels of genes are northern blots. And there's a number of different technologies which are kind of replacing the northern blot, but we'll go with this since it's a very standard sort of technology, and it's a good intro for understanding things like microarrays, which we'll talk about in a minute. The basic idea with northern blots is that we're going to separate the RNA by size, and then we're going to be able to visualize individual messenger RNAs by using a radioactive probe. So in a nutshell, RNA samples, so you take your samples of interest, whatever type of tissue you're interested in, brain, liver, skin, you grind it up, purify the RNA, and then the RNA is loaded into the wells of a gel and placed into an electric field. And basically, RNA, like DNA, is going to be negatively charged, so it's repelled from the negative electrode, attracted to the positive electrode. So the samples then will migrate through this gel. And this gel is a lot like something like Jell-O or something like that. So you can think about this as just sort of a mesh that these molecules have to work their way through as they move towards the positive electrode. And not surprisingly, like going through a mesh, the smallest things can get through faster than the larger things. So in a gel like this, the smallest things end up furthest from where they started, and the largest molecules end up back here. So what they've drawn here is kind of this big blurry line is kind of representing all the RNAs. So again, there's probably like 10 to 15,000 different RNA molecules in this sample. So you're not going to be able to see lots of individual bands. The couple of bands I have drawn in here are just are not real, but kind of virtual markers, just to indicate a few specific transcripts in there, so that you can kind of keep track of what's going on with them. So once these things are separated in the gel, the next part is that the RNA needs to get moved out of this gel onto a membrane, typically nylon or nitrocellulose. And practically speaking, this just kind of looks like a piece of paper. And that's kind of this very light rectangle here. And there's some various techniques that we don't need to get into where you can transfer the RNA from this gel onto this membrane. And the essential idea here is that it's moved kind of straight up and out of the gel onto the membrane so that everything maintains its position. So the same place where these bands were on the gel, they are now in those same positions on this membrane. And then once the RNA is immobilized on the membrane, we can use radioactive probes. So we can take a strand of DNA or RNA that's complementary to these RNAs, make it radioactive, and allow it to hybridize to these filters. And basically, the magic of this is that through that specific base pairing, these radioactive probes, which is made to be complementary to just one of those 25,000 genes, will find its match on this membrane, hybridize to it, and give you this radioactive signal. So again, this sort of this lane showing you this wild type, mutant 1, mutant 2, is full of maybe 10 to 15,000 different RNA molecules, but this radioactive probe, because it's only complementary to one of them, gives you a very specific signal. Now, some of the things to keep in mind with RNA blots is it's giving you quite a bit of information. So it's giving you information about the size of this messenger RNA that you're detecting. So again, things are going to move faster fragments move towards the bottom of the gel quicker. Larger fragments will be retained more towards the top of the gel, so you're getting information about the size of this RNA molecule. And also, the intensity of this is going to give you some information about the abundance. So in this case, wild type gives a certain amount of signal. This mutant 1 shows less, so this might be some sort of a hypomorphic allele where you've knocked down the expression. Whereas mutant 2 shows no signal whatsoever. And this could be due to something like a deletion, where you've completely eliminated the function of the gene. So northern blots are very standard techniques for assaying gene expression. The downside is that this is a sort of one gene at a time type of assay. So for each gene you want to look at, you make a radioactive probe which is going to light up just that one gene that you're interested in looking at. But ultimately, you'd like to know what's going on with all of the genes in the genome. So rather than just going one gene at a time, you need some sort of a way to scale up things so that you can look at the expression of many genes at once. And so if you want to look at all 25,000 genes in the genome, it's not very practical to make 25,000 different northern blots with 25,000 different radio-labeled probes. So basically what the solution is to basically reverse the situation with the blots. So in blots, we run out all of the RNAs in a lane of a gel, and then we make one probe, which will be specific to one gene. So what I'm going to show you in a second with microarrays is what we do is we take those probes and put those on our substrate. And then what we use as the radioactive part of this is this total RNA. So what's a microarray? So a microarray is basically a solid surface, usually glass these days, that have different DNA molecules laid down in a very regular pattern. So in a sense, what this could be for doing expression arrays, looking at RNA levels, is every one of these spots would contain a single-stranded piece of DNA that's complementary to one of the genes in the genome. So gene 1, gene 2, gene 3, gene 4, gene 5, the whole way through all 25,000 genes in the genome. So that every one of these spots will contain a DNA that will specifically hybridize to one of those 25,000 genes. Then the labeling process, again, it's much like northern blots, but a little bit backwards. So instead of labeling one probe, what we're going to label in this case is all of the RNA. So we take our samples, and these could be, say, a tumor versus a normal tissue, maybe tissues that have been treated with drugs versus no treatment, or if you're interested in something like development, you could look at embryonic stages versus adult samples. Isolate RNA from these, and then these are converted into cDNAs that are labeled with fluorescent nucleotides. So, and we do this in two different colors. So we add a green fluorescent nucleotide to one of the samples, and a red fluorescent nucleotide to the other sample. So what we've done now is we're labeling all of the RNAs that are in these populations. So all 15,000, 10,000, 15,000 RNA species are all now fluorescently labeled with either green or red fluorescent dyes. And then these two mixtures are mixed together and hybridized to one of these DNA microarrays. And again, each one of these spots contains a probe that is going to be specific to one of the RNA species in the genome. So this gives you something like this is what one of these things actually look like when you label them. And to interpret these, we look at the colors. So because we've labeled one of the samples with red and one of the samples with green, then if that particular probe is present at the same levels in both samples, say wild type or your mutant, your tumor cell versus your normal tissue, then these kind of average out to give you yellow. So that's why most of the things are yellow on this microarray. Now you will notice that some spots are very bright, like these guys. Some are very dim, like these. Some are completely black. And this, again, is giving you an indication of how highly expressed these things are. So in these particular tissues, genes like these over here are maybe not expressed or not highly expressed, where other ones are highly expressed. And the ones which are yellow, because these are roughly averaging out, means that these are genes which are expressed at about the same level in both tissue types. However, you will notice that there are some which are labeling as green. And what this means is that there's relatively more of this RNA in the sample that you labeled with the green dye than in the one with the red dye. So we have much more red than green. And then similarly, we can find ones that go the other way as well. The things which are relatively upregulated are expressed at higher levels in the tissue that was labeled with the red dye than the one with the green dye. And by doing these sorts of microarrays, we can look at all 25,000 genes and find those particular genes which are up or down regulated in the samples that we're interested in. And this gives you a handle on what really is the difference at the gene expression level between different types of tissues, be they tumors and wild type, or between embryos and later stages in development. So just for a second to talk about some of the problems and limitations with microarrays. So this is a really good technique and still gets used a great deal in biology. But it does have some limitations, and just like the EST libraries we talked about before, you still have the problems of detecting rare messages. So if something is just not present very often in your sample, then it's not going to label very much and won't give you much signal to hybridize to the microarray, so you won't be able to detect them. So you're still stuck with the rare things. The other problem is making specific probes. So this whole assay is based on hybridization, and it's based on the concept that a specific RNA, one individual RNA, is going to hybridize specifically to one of these spots that has a complementary piece of DNA. A problem with this is that, as we talked about earlier, that many genes are highly regulated to each other. So if you look at like these C2H2 zinc fingers, there's over 700 of these things, and they're going to have fairly similar sequences. So in some cases, it's very difficult to make specific probes which will hybridize to only one of these. So even if you try your best, a sequence might not be totally specific and might hybridize to, say, 3 or 4 of these 700 instead of only one, in which case this is going to cause you to lose some resolution in the interpretation of microarrays. Okay, so that wraps it up for this lecture.